some of the evening hour-based check-ins that we had. It was fun to see how everybody was doing. I'm excited today to hear your stories, to share with us and your reflections about your experiences with the microfauna from Montbrook, not just for you, but with your students. And of course, since this is a learning activity, I hope that you and your students learned lots. And I'm interested to hear what you did learn, how they learned. Nothing is ever perfect. This is our first time we've done this, sending out micro matrix with fossils in them by the mail, and then do this video conferencing COVID restricted kind of exercise. And we would like to understand how we could improve on the program that we've designed for you this time. So if and when we do this in the future, we'd be better poised to have a more successful professional development series for the next cohort of teachers. You should know now that you're part of our team, and you've connected with, I'm presuming that you have connected with at least one of our scientists, and you should feel that you now have a scientist-teacher partnership available for future collaborations. If you want to talk to any of us or your particular scientist that you were paired with, you should feel comfortable doing so now because you're part of the team and we're available for continued collaborations. We'll talk later about how you can continue to stay involved, but of course, I presume that most of you are signed up in our teacher network, our CEF's teacher network that Stephanie manages, mostly Stephanie and Brian, I guess. And if you're not, I think you are, but if you're not, you need to get in that network so you can get my monthly newsletter about what our upcoming activities are. So again, welcome, and I'm looking forward to a productive wrap up of this professional development session. And with that, I'll give the floor back to Brian. Thanks, Brian.
Thanks, Bruce. Appreciate it. So moving forward, talk a little bit about what today is going to look like. It's going to be a little bit similar to last time at the beginning in that we're going to talk a little bit about what some of our graduate students are working on, what their research is, how it impacts, society and the bigger implications of their research, and how that may be of interest to you. Just like last time, we'll hear from 2 graduate students, and then we'll move on to a Q&A session for them. Again, very similar to last time. Take a little bit of a break, and then we'll turn things over to Richard to talk to us a little bit about what do we do with the materials that were mailed to us? How do we return those to UF? Things like that, and what the logistics of that look like. Then we're actually going to turn it over to all of you. We're going to essentially have a very quick lightning talk from all of you. We're going to do a more in-depth session within our grade level bands, whether it's elementary or middle. However, before we do that, we want to get a chance to hear from each and every one of you. So we're going to do a very, very quick overview of your experience. You could share what your students experienced. You could share what you experienced, what you learned from being part of this. Whatever you'd like to, we just want to hear a little bit about what you did. And we want all of us to hear have a chance to hear from that and experience your story, essentially. Then, like I said, we're going to dive deeper into that idea, and we're going to take a longer period of time within those grade level bands. We're going to talk more in depth about what your activity was, who the scientist was you met with, how did that experience go? Things like if you were to do it again, what would that look like? What would you change? And those types of ideas and concepts. From there, take another break, and then we'll turn things, but we'll talk a little bit about how to stay involved, take a feedback survey, and begin the process of wrapping up this experience, this Paleo teacher experience. Oops.
Can I just jump in, Brian, that the survey is really important for us, for the teachers. We want you to know this because we're actually funded on this project by the National Science Foundation, and we need to submit a report on this as one of our activities for outreach for what's called broader impacts. So it is really important for you to participate in the feedback survey, please.
And our goals for this webinar, just to lay them out for you, we hope that you have a chance to learn a little bit more about Florida's paleontology content. That'll primarily be based between our scientist conversations in the afternoon, as well as hearing from our graduate student presentations. We hope you have a chance to reflect with your cohort on your activity, hear from other ideas if you were to do it again, and we hope that you do next year. And you have a really great chance to talk and brainstorm and build upon each other's ideas. And lastly, understand how to stay involved with the STEPS program. As Bruce said, now you're part of this big network, and we hope that you take advantage of it. Okay, so that being said, we're going to turn things over to Mackenzie. Mackenzie is going to be our first Spark speaker. She's going to give our first presentation on a little bit about what she does.
Awesome. Thank you, Brian. I'm going to share my screen, I guess. Yes. Okay.
Yeah, sorry.
Sorry. No, you're good. I think I took it away from you, but and get started. Hi there, everybody. For those of you who don't know me, my name is Mackenzie Ross. I am a master's student here at the University of Florida. I'm wrapping up actually my first year in the graduate program. And I'm really excited to talk to you guys today about my research. My research centers around the Peace River, but it really focuses mostly on science education and public outreach. And it's really inspired by SEFS and the SEFS program. And just a little bit of background on with me and SEFS. I actually started volunteering with SEFS as an undergraduate at UF. So I've been doing this for I think 2 years. And I tried to count the other day how many visits I've done. I think it's like 25, but I'm not really sure. But I've done quite a few, and I've worked on a couple professional development workshops as well that really inspired my research and my kind of combination of Peace River paleontology, science education, and public research or public outreach. So I'm just going to get right into it. So a little bit of background on the Peace River. I'm trying to minimize so I can see this. Here we go. So my thesis project is part of something called the Peace River Paleo Project. I actually have this linked in the presentation, and we actually have a website for it under the Florida Museum of Natural History if you guys and your students want to check out this project as a whole. And it's a multi-year effort. It's actually also with Dr. Hulbert, who is here today, and a couple of other people to study the vertebrate fossils of the Peace River and its associated tributaries. And the Peace River in this map is the area highlighted yellow, and it kind of starts in the bottom of Polk County and runs all the way through to Charlotte Harbor. But most of the fossil collecting is actually done in Hardee and DeSoto counties. And my aim for the first part of this project is broken up into two portions, the kind of paleontology heavy portion and then the science education and outreach heavy portion. So the first part, I'll be conducting a multi-proxy analysis of the diet and paleoecology of the upper Peace River fauna, specifically the herbivores. And that's a bunch of big fancy words. And basically all that means is that I'm using 3 different types of analysis, which I'll talk about in a second, to study the diet of these Pleistocene herbivores, maybe the environment they lived in, the climate they might have lived in, to build a broader picture of this area of Florida. And I'm doing this for a very specific reason, which I'll talk about on the next slide. But the 3 types of analysis I'm doing are microwear, mesoware, and stable isotope analysis, which will help determine if these herbivores are grazers, browsers, maybe fruit seed eaters, and whether they're consuming C3 or C4 plant material. And a little bit of detail into that, nothing too crazy, so I don't bog you guys down. But these 3 analyses are using physical, so mesoware and microwear both studying the physical impacts on the teeth, and then the stable isotopes actually studying more of isotopes or chemical impact on these specimens. So for the mesoware, we can help determine if a herbivore is a grazer or a browser by looking at how much wear is done on this teeth. So how much the teeth is actually worn down. So over here on the right, we have a picture of what that might look like. So an animal like a grazer that's eating a lot of low-lying plants, and those plants might have a lot of dust or grit on them. So a lot of stuff that might actually wear those teeth down over time. Might end up with a tooth like here we have in the bottom E that was really worn down and really flat. Whereas a browser that might be eating softer leaves, higher off of the ground, not as much dust or grit on them, might actually retain a lot more of those ridges and valleys into their old age. They might have, maybe middle teeth or pointier teeth, depending on what they're eating. And we study this actually just by looking at, as you can see, this kind of side view of this teeth. Just like this, and kind of rating it on a number scale, so 0 to 4 in this example here. For microwear, I don't have a very good picture of this. I kind of searched around, but I couldn't find any great ones. It is just studying the pits and scratches on a teeth. So how the food an animal's eating, usually in like the last 2 weeks of their life, actually pits and scratches on their teeth. So how it affects the kind of top surface of their molars specifically. So a browser might have a high number of pits, a relatively low number of scratches. whereas a grazer might have a high number of scratches and a low number of pits. And we have a way of kind of quantifying this and analyzing it. And then frugivores, so those eating maybe fruits or seeds or something like that, tend to fall in between these two end members. And then finally, for stable isotope analysis, which I've misspelled here, pardon that, for a C4 diet, these animals are usually eating trees, shrubs, cold season, temperate, high latitude grasses. For a C3 diet, they're eating bushes, shrubs, and trees. And we determine this by looking at carbon isotopes for the food and then oxygen for the water that they are drinking. And this bottom picture here is a picture of me looking a tad disheveled because I've been doing this for several hours, sampling a taper tooth, a taper molar, for stable isotope analysis. And we do this with a very small little hand drill that we kind of operate with a pedal. So it's a little bit of a fine art. And we scrape off the enamel, specifically the enamel of these molar teeth, into a piece of whey paper. And this portion of this project is actually what I'm hoping to use for my outreach portion. I'm hoping to actually make myself working in the lab, actually taking the samples of these teeth accessible through virtual paleontology to students in K-12 classrooms. So why am I doing this? Well, one of the reasons is that there's a very large hole in Florida's fossil record. So the Peace River has actually been collected for well over 150 years, but most of this collecting is done by amateur or hobbyist fossil collectors and not really professional scientists looking to do studies on the Peace River. There are a couple really good pioneering works on the Peace River. These, as you can see, are from the 1800s and are a little bit outdated with some of the terminology they use. And there's really no true descriptive review or comprehensive kind of research study of what we are calling the Peace River Basin, or the PRB. So the aim with this project is to kind of fill in this hole. And this figure on the right is actually from a paper that Dr. Hulbert found, showing some of the fossil sites from Florida. And as you can see here, where the Peace River is, there's a big giant hole. We have no real diet or geographic data here from this, these Pleistocene aged fauna. So the goal here is to kind of publish this research and continue to work with the Peace River Project to fill in this hole in regards to the Peace River area. My second portion, this is the portion that is probably a little more interesting to you guys, is my broader impacts deliverable, or my science education and outreach portion of it. And my goal with this second portion is to utilize the data and the data acquisition process from the first part and actually collaborate with a Florida teacher through the SES program to design a lesson plan. This lesson plan will be aimed for 8th through 12th grade teachers, but one of my goals with this research is to make sure it's reproducible so it can be reproduced at different grade levels as well. And some of the physical educational outreach portions we want to implement, as they've been so successful, as we saw with this mob book, is the fossil specimen kits. So the things that your students actually pick through, as well as scan specimens, which the SES program has used in the past. So 3D PDFs or 3D printed fossils, as they're a little bit cheaper and a little bit more durable for students to handle as well. And then the data gathering process, we really want to ensure is open access and accessible to all students, as I really want to focus on geoscience accessibility. So regardless of learning ability or anything like that. And we are aiming to determine if accessible scientific research at a K-12 level will increase interest in paleontology and earth science. And we will be determining this through both an educational evaluation and pre and post surveys of the final lesson plans. And these next two slides are just the standards. So these are the standards for my study. These are mostly middle school standards. So any of the middle school teachers in here, you might recognize some of these. So these standards that you guys have been using in your SEFs visits, any of the ones that any of our scientists kind of met for you guys, are the ones that I'm aiming to kind of encompass with my study as well. And this was actually inspired by a lot of the elementary school learning standards and a lot of my work with elementary school SEFs teachers, because most of these SEFs visits I do are elementary and middle school. I really don't do that many visits with high school teachers. So a lot of these learning standards that you guys are meeting in elementary school are just introductory learning standards to these higher level middle school and high school ones that I am aiming to meet for my study. And then this, I'm just kind of wrapping up here. This is my SEFs experience. Like I said, I've done quite a few SEFs visits over the past, I think, 2 years, both in person and virtual. I've worked on several professional development workshops. These two here on the bottom right are actually from my visit with Monica, talking about the Montbrook fossils and some Florida geology as well, which was an awesome visit. So I just want to put those pictures in here as well. And then finally, to kind of wrap up a little bit here, my research has really been inspired by SEFs, inspired by the teachers that I've worked with. As in the past, my SEFs visits have focused largely on Florida geology and paleontology, which is really a natural segue into the education and outreach portion of the Peace River Paleo Project. And here in the bottom left is just what a picture of the Peace River looks like during optimal collecting time. So it's usually around the knee level of water. And these are, as you can see, families and kids, sometimes class groups, fossil clubs that go collect at the Peace River, which is one of the reasons why I am so happy to be doing research with the Peace River, because it's not this big kind of scary, fancy professional scientists like collecting site. It is a site that is predominantly collected by people just like everyone, you know, families, teachers, students. So I think it's the absolutely perfect site to be doing scientific education and research, or sorry, outreach research. And then finally, my major focus for all of this is virtual paleontology, which I've said a couple times, which really draws on the ideas of citizen science and community engagement. So similar to a SEF setting, the students will actually be able to experience me doing lab work through live Zoom-in chats, live Zoom-in calls into their classroom, and watch the process of data acquisition and analysis, but at their learning level, but for a publishable study. And we will be combining this with in-classroom paleontology teaching aids to foster both excitement for Florida paleontology, but also really teach them a broader understanding of why we're studying paleontology at all grade levels. That is all I have for you guys. Thank you very much.
That was awesome, Mackenzie. Thank you so much. And so relevant in so many ways. I can speak personally as a former teacher in bringing students and families actually to the Peace River and having some of the graduate students at UF's paleontology and geological sciences departments come and work with us and collect. So it's a really good experience for all, and certainly something that the kids and the parents even remember and have come back years later and mentioned, or something comes up on their Facebook memories and feeds and they'll reach out to me and say hi. So it's a really nice time. Mackenzie, I had a question for you, and I'm going to start with mine and just see if anybody else has any questions before we hop into our next spark real quick. How do you differentiate between the microwear and say just weathering and erosion that happens on fossils as they're kind of being pushed around over time? How does that differentiation happen?
So that's actually a really good question. We have to be very careful with the teeth that we're picking. So that's one of the things I'm doing right now is I'm actually working with Dr. Hulbert to go through all the teeth in our collection to kind of determine which ones are okay for like this microwear and these mesowear studies. Because a lot of them are too long, because it is a water environment. A lot of them have undergone a lot of water erosion, a lot of stuff that has just kind of worn away all of those pits and scratches that we can study. So sometimes you have to take a look at them under a microscope and really just kind of suss out whether it is an appropriate tooth for study. And a lot of them, like I said, we can't even use because of the environment that they're in. They've just been too worn away. So that's been a fun thing doing, is figuring that out.
Yeah, thanks for answering that. Does anybody else have a question for Mackenzie? And if you do, we'll come back, bounce back. But I figure we'll roll into the next one. We have Ms. Jeanette Perlow on with us today, and she's a PhD candidate who has done work extensively in the Montbrook area. And I'm going to let her dip in and tell you a little bit more about her research and what she does. So Jeanette, take it away.
I'm going to stop your screen share stuff. Hello, everyone. I'm sorry, my camera has to be off because my internet hates everything about my house. You should be able to see my presentation. Okay, so hello, I think we've met, but if not, my name is Jeanette Perlow. I'm a PhD candidate working with Bruce and at Montbrook, the Gomphotheres and Mastodons, and I think my internet may have cut out, so if it did, I'm very sorry. Please bear with me. I don't know why it's being so difficult today. And I come from California, and I had no interest in paleo until I met Bruce. I actually love telling the story, and I think he might actually hate this story. But I found paleo to be very boring, and I wasn't a fan. And then Bruce brought me out to Montbrook, and I found my first turtle, and I was hooked. And I was conspiring to figure out how to convince him to let me dig for the rest of my life. And then he asked me to be his grad student. And long story short, here I am 5 years later. And now I absolutely love paleo, and I'm very glad that this is my career. So I've been working for Bruce for the past 5 years or so, and I've had a chance to dig not just at Montbrook, but in Nebraska, and in California and Panama. And it's been a really fun experience, but most importantly, it's been a learning experience, and it's been a way to connect with different communities, not just local communities, but also educators, students at different levels, and the entire process from teaching to planning professional developments, to disseminating the information. And so it's been a lot of fun. All right. So moving forward, a little bit on elephant evolution. So in the group Proboscidea, you have, it used to be this really huge group of animals. Now we only have 3 extant living individual species: the African bush elephant, the African forest elephant, and the Asian elephant. But prior to that, there were many groups of different types of elephants. And in the bottom pictures, you can see one of my favorites is Dinotherium, which had these long recurved tusks coming out of its lower jaw, but it didn't have upper tusks. Gomphotherium, which is a fun group to look at. It has 4 tusks, both upper tusks and lower tusks, and some of them you might know as shovel tuskers, where they have very wide lower tusks, and they're usually depicted shoveling muck out of marshes. We don't believe that's how they were used, but they're a fun group to inspire curiosity about what these elephants were doing. And then the famous mammoths and mastodons with their massive tusks that lived into past the just a little bit after the Ice Age, and early humans were able to live among them, and hunt them and eat them. So I study the group Gomphotheridae. And elephants migrated into the Americas about 16 million years ago. There was 2 instances of migration into the North American continent. They began in Africa and moved into North America about 15 to 16 million years ago. And then they migrated south into South America. And so elephants and elephant relatives can be found all over the world except Australia and Antarctica. And this map shows the movement of mammoths. So it's not pertaining to Gomphotheres, but it's very similar to how other elephant groups moved into North America. And I really enjoy it. So the dates shown on the map correspond to mammoths and not to Gomphotheres. Gomphotheres were some of the first elephants in North America. And the chart down below shows a current tree of how the Gomphotheres in North America and South America are broken up, with Gomphotherium and Rhynchotherium, which we believe may be at Montbrook, the Rhynchotherium, and then Cuberonius and Nodomastodon are the usual suspects in South America. Excuse me. And so elephants, apart from their really cool tusks, they have these really unique, massive teeth. And I want to show you just the diversity in tooth shape within the elephants that I showed you on rhino-like or taper-like teeth. Mackenzie, I'm sure you'll recognize the shape a little bit. It's very taper-like, but larger. Gomphotheres also have these chunky teeth here in the center, and you'll see another picture in a couple slides. Mammoths have these massive, massive teeth, and you can see that the shape recurves, and that's because of the manner in which teeth erupt in elephants. So like sharks, they have horizontal tooth replacement, in that the teeth cycle forward in the jaw, except that they don't replace their teeth throughout their lifetime. They just have, depending on the species, about 6 to 8 teeth throughout their lifetime. And that is great because it helps us age the individual animal. So if we know what tooth is present in the jaw and how much wear is present, then we can determine how old the individual was at the time of death, or how old the individual was of that loose tooth that we found. So that's a really cool way for us to ID the age of the animals. And in the top right corner, you can see that the African and Asian elephant teeth. And looking at just the surface of the Asian elephant tooth, it looks very similar to the mammoth tooth. And so we use the shape of the feeding surface, the feeding portion, the occlusal surface, to help determine what animals we're eating. Are they carnivores? Are they herbivores? Do they eat grasses or leaves? So are they browsers, or are they grazers eating, are they browsers eating leaves or shrubs, or are they grazers eating grasses? And that's, prior to any chemical analysis, we can look at the surface of the teeth and have some general idea of what they were eating. So moving on to the Montbrook site, we have over 30 jackets collected of skulls or mandibles of elephants. And we know for sure that we have at least one gomphothere, which may or may not be a rhynchotherium, and perhaps a second species of gomphothere. And then we also have mastodons at the sites. And so as you remember, the site's 5 to 6 million years old. And I really like this picture, this artist's depiction, even though it's not based on the Montbrook site. It has a lot of the animals that are present at Montbrook. So we have a lot of the waterfowl, a gomphothere, there's a gator in there, a rhino, some tapirs, otters. And I really enjoy, every time I look at this picture, I see another animal kind of secreted away. So I really like this picture. And so with the gomphotheres, they're characterized as having a long, low skull. So when you think of a mastodon, if you've been to, or excuse me, a mammoth, if you've been to the museum here in Gainesville, the main skeleton when you first walk into the hall is a mammoth, and it has this really massive, tall head. That's not gomphothere skulls. They have a much lower, low skull. Their tusks, their upper tusks have this enamel band, and you can see this in the picture at the bottom. This is dark brown yellowish material. That's an enamel band that runs along the surface of the tooth. So like our teeth, which have enamel on the outside, elephant tusks don't normally have that. So modern elephants, the ivory that we see is the dentine, which is a softer material, but different groups of elephants did have this enamel band that, and Stephanie, stop me if I go over time. I stop paying attention to the time. They have this enamel band that spirals along the length of the tusk in the upper tusks, and that can be used for chemical analysis as well. Additionally, they have a elongated lower jaw to accommodate the lower tusks that are in it. And I really like this upper picture that shows about how far the tusks go into the lower jaw, as well as the teeth cycling in from the back of the jaw and moving forward. And they do have this downturned elongated mandibular symphysis, which essentially means they have a really long downturned chin. And this area right here, you can see where it kind of is really long and downturns, and that's an elongated downturned mandibular symphysis, where the two halves of the mandible species, and that's a big portion of what I'm doing to figure out which species are at Monbrook. So a little bit on my current research. On the left, you can see all the jackets that we bring back from the field that need to be prepped out. And they sit in the loading dock until we can get to them. So they're a fun mind experiment. When you need to get away from computer work, you can just prep. But I'll be doing species determination and description of the elephants at Monbrook. So are they Rhynchytherium, or what else is there? I'll also do, like Mackenzie, stable isotope analysis and microwear analysis to understand how that ecosystem was partitioned among the mega herbivores. So like I mentioned, we also have a mammoth, we have rhinos. So how is this ecosystem being shared among all of these large herbivores? And my population dynamics chapter will look at how many species of proboscideans were present, how the bone bed was accumulated. Did it happen overnight? Did it happen, over a long period of time? So was it catastrophic? Was there some event that caused a mass die-off? Or was it a slow buildup of a bone bed? What was the population structure of those animals? So was it a growing population of elephants? Was the population already in danger of not surviving because there weren't enough young being produced? And how big were these proboscideans? So establish an estimate of body size or body mass for these individuals. So a little bit on species determination. One, I love this picture, so I feel I need to share it anytime I can. But you can see the downturned symphysis here. This is a lower jaw with a downturned symphysis, or the chin. And you can see the angle, but using both the downturned symphysis and the shape of the jaw and the tusks and the amount of spiraling of the enamel, plus some other morphological features, I'll be able to hopefully, fingers crossed, once I compare it to the type specimen, which is essentially the definition of that specimen, determine which species we have at Monbrook. For the stable isotope analysis, similar to what Mackenzie is doing, I am drilling the teeth for enamel, and the top picture is a picture of a Gomphotherium mammoth. Oh my God, words. Gomphotherium molar. And you can see that it's very bulbous and has all of these ridges. So that helps us determine that this animal was most likely a browser. But to really figure that out, we take small samples of enamel off the teeth. And here I'm doing what's considered a serial sampling. A bulk sample, I would just take a sample anywhere along the length of the tooth on the enamel and use that as the average values for diet. But with the serial sampling, I'm taking samples at, in this tooth in particular, it's every 5 millimeters to determine if there is a shift in diets from when the tooth was first mineralized, so at the tip of the teeth mineralized first, to when it was finally mineralized down at the bottom closer to the roots. And so along the way, I hope to find different values, perhaps see a weaning signal when the animal stopped consuming milk from the mother and went on its own diet. And I also wanted to show just the little amount of sample that we collect per vial. We need at minimum 2 milligrams of sample after it's been pre-treated, and then it's ran through a mass spectrometer. So essentially they just quickly combust the powder and capture the CO2 that's released. And those are the values that they give us. And the plot on the right shows a quick plot of half of the samples that I've collected thus far. And we can see that they're kind of scattered between 0 to -10 in the carbon values. So these are delta 13 carbon values. And the more positive side, numbers that are closer to 0, tell us that these are probably grazing animals, whereas the negative numbers, the ones that are becoming more negative, are telling us that they're more browsing. So that is one way for us to determine, are they C3 browse eaters, so the more negative numbers, or are they C4 about temperature and their water ingestion. I see that my internet may have cut out, so please let me know if you guys have any questions. I added this slide last minute because I didn't want to take from Mackenzie's microware study, but I wanted to show the different textures since that was brought up. On the left, you have a traditional microware analysis where you looked at the different pits and scratches. So you can see the scratches here on the top, say the first 3 panels, and then the pits in panel F and G. And so depending on the amount of scratches versus pits, you can determine, were they eating more browse, were they eating more grasses? And that's the traditional microware. The one I'm working on myself is more of the dental microware texture analysis, or DMTA. And this, we take a resin cast of resin mold of the tooth, just a section, and then it's cast with another material later, and it's then analyzed by, you analyze the 3D surface model of that cast with computer software, and it creates this 3D visualization of where pits and scratches are, and from there, based on that, we can determine, what were they eating. And so that'll work hand in hand with the carbon and oxygen values to better determine what these individuals were eating and how the vegetation was being split up between all of the herbivores. And finally, my population dynamics chapter will look at the structure of the population at Montbrook, how the herds were composed, how old they were, how big they were. I like having these 2 pictures here because the one on the left is an adult Gomphotheres, and the picture on the right is a baby mastodon with a little tusk kind of peeking out of the lower jaw on the left. And I love these 2 specimens. I think they're great engagement tools. And then one thank you to everyone that's helped out with Montbrook, but yeah, if anyone has questions, please.
I believe we have a couple actually. Eusebius and Monica saw questions in the chat box. Do you want to each hop on and ask your questions?
Sure. Mine was very basic, and I apologize if I should know it, but just the definition of a jacket. I was trying to Google it, didn't come up with anything relevant, but how do you define a jacket?
Yeah, so that's a really great question. Thanks for asking that. So the jacket itself is made out of plaster material. So like when you break a bone and you go to the doctor and get a cast put on, it's essentially the same material. Either for the really big things like the skulls, we use plaster of Paris powder mixed with water and burlap sack strips to create the jacket. So essentially it is a protective sleeve, we can call that, or a protective cast around the bone that is still in the dirt. So we make a mound of dirt around this bone and then wrap it with the wet plaster material and burlap sacks, wait for it to dry, flip it over, and then carry it back, bring it back to the lab. So you can call it like a protective cast or protective sleeve so that we can prepare the bone in a really organized manner and a controlled fashion where we can slowly clean it without worrying that some of the pieces might fall off.
Thank you.
Yeah. Other questions?
Yes, my question was, when you're looking at I saw you were looking at the different teeth and how the different teeth kind of wear down. Did you see any kind of change as the environment changed with the wear on the teeth, or they it just all depends on what they were eating. They have like they have a certain type of wear on their teeth. That means, say for instance, if they was eating soft, eating stuff out of water and stuff like that, did their teeth remain the same?
Yeah,
Or
so
do you
that
see changes when you're looking at as far as the age of the elephants?
Jeanette, you faded out there for a second. I think you're answering the question.
okay, can you hear me now? Okay, so I think there's two parts to that question you're asking. Can we see within the age group as the teeth are wearing if we can see different wear on the teeth as they get older, and also what that wear looks like, I think is what you're asking. So we haven't finished that portion of the research. We are starting that, or I'm starting that in the next couple of weeks with the microstructure. Regarding characterizing the teeth, it's hard to see how much it changes, like those micro wear. That microwear evidence is usually seen through a microscope, so it's really hard to see that with the plain eye. So right now, we don't have an answer for that from Montbrook. Keep in touch over the next coming months, and we will, hopefully. But you would see, in theory, a difference in wear on the teeth microscopically. So if you look at the really young baby teeth, where they're probably not eating a lot of vegetation, most of their food is coming from the mother's milk, you should see less pits and scratches. And then as they get older, you would see more pits and scratches as they're eating more vegetation. And then depending if they're migrating in and out of the Montbrook area, you might see a difference in that. But one thing that we need to remember with the microwear analysis is that it only gives us the last couple weeks of diet that the individual ate before the tooth was buried. So we probably wouldn't see a huge difference from one individual to another, because migration takes so long to move from one place to another, and they're eating along the way. But there have been other studies where they have seen that regionally, different mastodons were eating different things, even though they're all mastodons and they're all browsers and they all live around the same general time. Regionally across latitudes, they're eating different things, and you could see that in the microstructure. So if we were to expand this study looking at Gomphothecus, say from the Tennessee gray site, we should see maybe a difference in microwear structure, since the Tennessee gray site is up further in latitude and in altitude, so the vegetation would be different.
Awesome.
That was a long one.
Great. Are there any other questions out there for either of the Sparks today?
And I saw the question about the PowerPoint slide. Yes, I can make mine available to Stephanie and Brian so that you guys can share it with your class.
Awesome. And we did stick the Google Drive in there once again. It's the same Google Drive that you all have access to. The link is in the chat, and that's where everything will be found. So you have it. I think Brian's going to throw up next, if there's not any other questions, a back in 5. We're going to take a quick break. So we will see you all at 10:50. And at that point, we'll have Richard kick some things off when we come back. Okay, see you in a few.
All right, welcome back. I hope you had a nice chance to stretch, get up, walk around a little bit, refill your cup with coffee, water, use the restroom, whatever you needed to do. Our next session to resume our plan for today is turning things over to Richard to talk a little bit about how all your materials are returned to UF and the paleontology department and what's next as far as the materials go. So Richard, over to you.
Okay, thank you, Brian. So really the most important thing to return would be any specimens that were picked out of the matrix samples that were sent to you that have the collector's data. This is not, some of you also got samples of matrix from the spoil piles. Those we are less concerned about unless you found something you think might be extraordinarily important or significant. And you could send me an image of that to confirm that. But for the materials that we sent you that had a collector's name and date and some square coordinates, we would like all available fossils returned to us, and they can be shipped, it can probably with the, inside the vials or plastic bags that you were sent. And we can get you the exact shipping address to me at the Florida Museum of Natural History. We are not concerned, again, you can keep the tweezers, the Petri dishes, any other supplies that you didn't use up, you're welcome to keep those. So all we're really interested in is picked fossils. If you have samples of the matrix that you were sent, again, this is the stuff that has the square data, that wasn't ever picked, or then you could also return that if you wish to. If you have the sediment that has been picked, so all the fossils have been removed, we don't need that. You can just discard that. That's about all I really needed to say about that. Going forward, when we return this material, it'll essentially just go into our preparation pipeline along with other picked specimens that I have volunteers and some museum staff do. And the next step will be to identify the specimens, comparing them to, we'll get you the address. I'll forward it to Stephanie and Brian, and they'll send it out to you. And so we'll identify the specimens. We'll determine if they are complete enough. I mean, some of them are going to just be broken fragments or really tumbled, water-worn, say sharp teeth or something that does not have scientific value. But everything that has scientific value that is from one of the squares with data will be identified, and then it'll be cataloged, assigned a unique number in our database, and then will be placed in our permanent research collection. As I mentioned to at least I think in one of the evening sessions, if you collected specimens from kids, from your students who made some attempt, and you made some attempt to retain which student collected which specimens, you could keep the student's name with those specimens, and we would be glad to sort of consider them part of the collecting effort of the specimen. And so they would, because they put in some effort to actually find the specimen. We would probably need to make sure that we have, because their name would then be published on our database, we would need to make sure that we had parental permission to place their name on the database, because that's publicly accessible. That would be the only information, would just be their name, obviously no other data about them, and no links to their identity, but just in case. Again, if you didn't do that, if all the students, if you just picked the stuff yourself and your students didn't do any of that work, or when the students return the specimens to you that it all got jumbled together, then obviously we can't do anything with that. But I just mentioned that because that was brought up at one of the nightly review sessions. I think that's all about returning that we need to know. I'd be glad to answer any questions that anybody has.
Awesome. So we will send an email follow-up with the address for everybody and just kind of reiterate those directions so that you have them. Brian, you want to throw up what's next? We have a question now.
There actually is a question, yeah.
Yeah.
From Anna Maria Schoonover.
Go ahead, Anne Marie.
Unmute myself. Well, I was wondering, it might be difficult for me. I did separate all mine and put all their names on it with the scientist's name. You know, like I had his stuff on here, but then on their back, I had all their name and school information. So now I told them that they're going to be, you know, recognized basically. I don't, but should it just say, can I just say Anne Marie Schoonover's class as on the database, or because I don't know if I'm going to be able to get permission that fast, you know, from all the parents. Like I have to go.
The way the software that we use, we can create a group so that it could have that name, you know, your name, class, even the school information, and then there would be a list of the individual participants that are part of that group. I believe that what would then go online would just be the name of the group. I think what you're asking is doable. So if you want to do something like that, that would be okay. So we would create, like I said, a group, and that name could involve the school teacher's name. It could involve the class grade level. It could involve the school name, and then I would also then need a list of the individual names to put in the database potentially. And that part would not be publicly available on the online database, only the name of
Bruce, I think we could hear Jeanette in the background there.
Were there any other questions, Brian? Or
Well, I guess my question is there any urgency? When would you like them back by? Is there an ideal time or date?
no, there is no extreme urgency. We have quite a backlog of material, so it's not, you know, the only thing would be if you think you found something significant, and I haven't had any, you know, dire email saying, you know, rodent jaw or anything like that. So I'm assuming what most people found is the ordinary stuff. So it's more, we're just trying to be, because we're excavating the site, you know, scientifically, and we don't want sort of a bias against a particular area in the site where we sort of taken data away from it. So it's just more a matter of us being sort of complete and having all of the data from the areas we're collecting this. So yeah, if you're still working on it, if you still have leftover sediment that you might just for fun and giggles want to keep picking at it over the summer, you know, there is no deadline like that.
Anything else? Any other questions? I'm sure there will be when the time comes to start returning materials, and that's fine. That's fine. Feel free to reach out to any of us. We'd be happy to help you with those questions. All right, great. Thank you, Richard. Monica, you look like you unmuted a second.
Well, it's somewhat unrelated, but my kids keep asking when they can show up in person at Monbrook. So just FYI, read it.
Well, you teach elementary, so it might be a few years.
Yeah, are we going to talk sort of about that next?
This afternoon.
Yeah, okay.
Yeah,
So
we'll
we'll
come back to
move that topic to a later time today.
All right. Moving forward, we're going to put you guys on the spot just a little bit. And this is going to be our lightning round, meaning it's real quick. We're going to give each of you maybe 2 to 3 minutes, and that's it. I'm not going to cut you off if you get excited. I love hearing about the exciting things that you're doing. However, this is just meant to be something very quick, very brief, just an overview of something that happened in your classroom. If you want to talk about your activity, that's great, and how it went, that's fine. Again, we're going to go into more depth after this session. We're going to go into that with your grade band. This is just meant to be very quick. Here's what we did. Here's why it was exciting. Here's what we did. Here's the scientist that visited me, and here's how it went. Something very quick like that. So I'm just going to go in order, and I'm sorry to put you first, Monica. but feel free to unmute and tell us a little bit about what your experience was like.
No problem. Question though, may I show my PowerPoint very quickly just to support?
Absolutely.
Okay.
Let me make you co-host, and then I'll just go through and make
I'll
everyone
give you like
co-host.
the shortened version of it.
Yeah, you should be able to share now. Go right ahead.
Okay, share screen. Yes. Share. Can you see it?
Yep.
And I want it in a slideshow, but the bar is blocking it. Oh my goodness.
You should be able to move that bar around. You can move it to the top or the bottom.
Oh my goodness, yeah, I can, yes. Slideshow from beginning. Can you see
There
the
you
full?
go.
Okay.
Sure can.
So this is just who we are. I have 22 fourth graders, so they're 10 years old. 14 of them are face-to-face, 8 are online. These were the standards that we focused on in the process. So just, from basing, these are the ones we discussed as a group, I believe, raising questions about the natural world, going over the tools, what does the information give us, making inferences, etc. And this is Kwong here in the picture. And this is my ELL student who speaks no English, and yet it was just amazing how he was able to, apply the new vocabulary and express what he's finding. In the process of it, so this was just the prep work. Just the conversation about who's a paleontologist, how is this person different from a geologist, anthropologist, or archaeologist? So we went over the Greek and Latin roots, affixes, etc. I felt like it was a good conversation that was related to the topic. And I have a few student samples that you see here. Of course, I showed them the video. They love the story of Montbrook. And again, like I said, they keep asking when they're going to show up and work with the scientists. Let's see. Also, we had this conversation, about just the tools. What is the difference between the field kit and the lab tools? This was the setup that I had in my classroom. I used trays just to keep everything contained. We had plastic cups, see-through cups. Next time I will have to buy more tweezers, right, to help them maneuver the fossils within the matrix. But this was basically what my students were working on, and it was awesome to have one-on-one. This is my Brooke here in the process of going through the matrix. Again, examples of students working. I provided them with the color copies of the PDF sent by you for the identification purposes. They also had laptops as means to refer to that reference sheet to help them identify the fossils. Here's our virtual visit with Mackenzie, which they loved, and they were very inquisitive, lots of questions about paleontologists, about becoming a paleontologist, about just the process of, the history of Florida, the geological history of Florida, and what the fossils mean, what they tell us. So thank you, Mackenzie, once again. They loved your visit. These were a few slides from Mackenzie's presentation, and she hit so many important points, including the geological history of Florida through, the most popular rocks we find in Florida, through springs, etc. So it was very enlightening for my kids. Mackenzie also spent a portion of her presentation helping us identify some of the samples my students found. So I took pictures of them, and then Mackenzie was able to embed them in her presentation and show us the visual support. So this was very powerful. Here's a recording of their testimonials. So I guess I will share it in our small groups, right, Brian, Stephanie? But you will hear them here sharing what they thought about the experience, why they thought it was important. So many of the responses were basically it was so cool to, first of all, to work with fossils from Florida, right? And then what it taught them about what was roaming in our state, what it means, what the animals ate. They also talked a little bit about the session with Mackenzie and how powerful it was for them to realize that it's not extremely, I don't want to say difficult to become a scientist, but it is within their reach. Some of them have this notion that it's not for them. So just being able to speak with somebody who can share, the obstacles they overcame to become scientists, and that you don't have to be perfect academically to become one, I think is very powerful for my students. And then I just have a slide with a thank you. Did I cover it all?
That's perfect. Thank you so much
Okay,
for
great.
sharing that. Yeah. Up next, we have Denise.
Good morning. I have, at the time when we first started this, I had 20 third graders, all virtual. And we visited with Dr. Maria. She came and joined us in one of our sessions. And I do have a slideshow that I'll share with the students' fossil findings in their matrix bags that they sent them. It was super powerful. One of the things that, hopefully if I can do this next year, I'll have them face to face and I can see their faces when they're digging through the little bags of fossils, because that we could see it online, but seeing their faces in person doing it, I'm sure is totally different. I had a student yesterday who were here for FSA testing, of course, and she was late and her parents were late. So I said, you know what, I still have bags that you can go through. And she sat and went through bags with me. And it was amazing to see her like make the connections of what she said. Dr. Maria said that this is the structure. That's how we know it's a bone. And I'm like, can you say that again, sir? I can record that. And she goes, I know it's a bone and it's not a rock because of the structure of it. And it was just for her, that connection. And it was a month ago when Maria came to visit with us. And so for her to keep that in her mind was super powerful. And for her, she only wanted to find the frog fossils. And I'm like, baby, you got a lot more in there than just frog fossils. Nope, that's all she wanted to find. And so it was amazing. And I cannot wait to do it again. And when I tell them that, because they all sent me pictures and I told them that I was going to share that with the group, and they're like, you get to see other scientists? I said, yeah, we get to see other scientists, and I'm going to share what you found, your pictures that you sent me, with the other scientists on Saturday. And so they're super excited. So they've all told me that I need to tell them about my class that I had on Saturday.
I love that. What a special experience. Thanks for sharing. Next up, we have Anne-Marie.
Hello. I have a presentation, but I'll save it. So I feel unscripted right now, but so my kids absolutely loved it. I think what surprised me the most is the quality of their vocabulary and their ability to use it after all of this. Some big words that as kindergarteners, I would have never really thought would have stuck, and they're just using these words. I was sitting here up until yesterday, doing fossils, and I'm just listening to them talk to each other, and they're using like, well, this one looks more porous and this one has, holes, and this one, just like big words that I'm like, oh my, like they're really making these connections and things like that. And, the fact that they were all very actively engaged because it was very hands-on, and even some of my lowest of my low, even one of my ELLs, he's not just ELL, but he's also in the process of interventions for ESC possibly. And even he was trying to say things, which is really hard to understand him just because he has like a language processing, thing. But when the kids would go, we like, I have it in my presentation, but the minute after we met with John, they were going outside on the playground, and the entire class, not just like a few, were out together digging and saying, we're the paleontologists, we're finding fossils. And he was right there with them trying to talk about it. Like, he couldn't say a whole lot, but he was trying. He was one that grabbed all of the fossil matrix and just threw it in the jar. He wasn't quite sure what to do with it, but he was trying. I like the amount of literacy I was able to do with my kids. There's a lot of really great books out there for young children about paleontology and fossils that I've now discovered. I also was very humbled by my expectations were not very high as far as what they could do with the matrix. And I kind of waited, and this is what I would probably do different. I waited a little bit too long to start the fossil dig. I was trying to like scaffold and build up to the fossil dig and really expose them to other parts of paleontology first. But the minute we were doing the fossils, I was amazed that they could start separating the types of bones. Like I was like just hoping we could just separate rock versus fossil, like, which is what we did. Some of their bags are not like all, one type of fossil. They're a mixture, but they were starting to mix them up. And like no, this is a bone, this might be a frog bone, or this might be this is a shark's tooth, this is could be a stingray barb. I was like, oh my gosh, I could have done better. I could have like actually had you guys have a list out, and you could have looked at it and separated it way better. So that's something on my part, I'm like, wow, they really could do more than we really gave them credit for. So I was really, really excited about that. And they absolutely loved seeing the scientist. I know John probably was like, I don't know how she does this with kindergarten, because their attention span's about 5 seconds, but they were very engaged. And I actually sent a picture of him with my kids to a friend of mine who's a science teacher at a high school. And she showed her science class. She's like, look how engaged these kindergartners are. Why can't you guys be this engaged? It was really sweet. But so I was really excited. They were excited. They've really grown a lot from this experience. So I'm glad we did it.
Thank you. I really enjoyed observing that one. That was a lot of fun. Susie.
Hi, everyone. So I just want to say that this tied in so well with the 7th grade standards. There's really so much that you could expand upon this, because I only touched upon like a small portion that it could tie into. So I think definitely to continue next year, it could add more to this, that will definitely cover a lot of the standards. The students enjoyed it so much, and I think that they will definitely not forget that, and especially the concepts that we talked about during, having such a great hands-on activity. My virtual students did come and pick up the spoil pile matrix, so that was awesome that they could participate with us, because that's really been a challenge to doing hands-on activities with the virtual students. They really enjoyed being able to ask the scientists questions, because the expert questions, because I couldn't answer all of them, so that was fantastic. I'm going to share some pictures with you, and I'm excited to know that we can have a little bit more time to look through the fossils matrix, because there's really still a lot more to go through, and we just need more time to do that. So hopefully I'm sharing this correctly. You guys see those pictures?
Looks good.
So far. So what amazed me was that kind of what Anne-Marie was saying was, they did better than I expected, and they really saw things so tiny. I was like, how did you even see that? So they got really pretty good at sorting things into the different groups. We were kind of trying to put all the, this fish scales together and the vertebrae together. I think it's, it's a little bit more challenging for them to really specify which species, it is in particular, but they were able to, like we put the bones together and all the vertebrae. And then I had like a digital microscope hooked up to my computer, so we could really zoom in on some of them and see like more closely, like, like the little barbs and teeth. And so that was pretty cool. But this is just kind of an idea of what they found. This was kind of random things. We weren't really sure where to put those. So that's pretty much it. But I'm excited about expanding the next year and adding to what we did.
I love that. I love that you're already thinking about what's next and how you could do it differently. Next, Eusebius.
Hello?
Hey there.
Okay, I'm making sure, 'cause, you know, I got a lot of madness going on with my kids in the background. But anyhow, so what I'm looking forward to, because scientists have been, we haven't really worked with the kids yet, but while the information about the kids going on through the spoil pile and stuff like that, it just changed their whole mindset about what's going on in the ground, in the world around them. And it's kind of, you know, being down here in South Florida where we got a lot of concrete and stuff like that, it just don't kind of give the kids a light of what's going on up underneath the ground and what has happened in the past. Because sometimes when we talk about evolution, natural selection, and we start talking about how things change over time, it's kind of difficult for us to go ahead and get the idea for my kids to understand that part. Because when they look outside, only thing they see is concrete. So it's a whole different mindset. It's a whole different mindset. So I mean, I'm looking forward to when the scientists come in the classroom because he's gonna help give the idea. Because now they ask the question about the particular little animals and stuff, but they try to figure out because they thought everything that lived a long time ago was huge. And I talked to them about the different fossil records and stuff, and I say, well, mammals at the time they weren't huge. But my kids, when you start talking about million years ago, 2 million years ago, they think everything was dinosaurs. So it has helped me kind of get a better get them out of that mindset, thinking about dinosaurs all the time. And that's it.
That's perfect. What a mind-blowing experience to finally realize that there are other things besides that concrete below you. Next, Jeffrey.
Hi. So, yeah, my 7th graders had a really great time going through all the matrix. I had the PowerPoint up on for them. They all had a copy of that so they could start to identify the different species or at least the different kinds of fossils. They did pretty well with that. It was funny. They were so excited that sometimes they would kind of skip that part. And I wanted to go to each student individually, and say, okay, well, this is this. How are you doing with this? Why don't you do this? They ended up lining up around my bench. And what's this? What's this? What's this? So they were very excited. Eventually, I was able to get them settled down and start doing their own identifications, and they did pretty well with that. They have their individual picks, and I have them in different little baggies, and that's how I'll send them back with their names on the baggies. It was very difficult to get them to stop. We had to get to some more curriculum, and they just didn't want to do that. But Susie is absolutely right about it falling right in with what we were doing. And I would really like to be able to do some more with that next year, maybe do it some better, maybe fold it into the curriculum a little bit more. They did learn a lot about Florida history with this, which was great. And with them working with fossils, none of them had ever done that before. I mean, they'd seen the collection that I have, and I usually use fossils during class for different things. But they told me that they had never realized that fossils could be so small. And I thought that was very cute. So they did very well. I was very happy with them. The applications continue to go on. We're working on natural selection and evolution now. So we can actually use our fossil experience for that. How did fish evolve in the last, 5 million years? Did they? Why are alligators still the same after so many hundreds of millions of years? So that's been pretty exciting for them. And they have like a better understanding of Florida history and where they live. That was one of the things that I hadn't really realized, is that these kids who have lived here all their lives have no idea where they live in such a wonderful and almost exotic place that they live in. So yeah, I want to do this again. I am still continuing to pick through the matrix. I found a couple of more neat things. I'll show the group later on. I think I even have a toe bone of something. So yeah, thank you very much. This is a good thing. I want to do this again.
Great. I love your enthusiasm. Yeah, Bruce.
Jeffrey, that was really cool. I want to tell you that the questions that your students are asking were the same questions that we've asked in our graduate seminar in vertebrate paleontology. We are wondering how long, why and how do alligators stay remarkably unchanged for so many millions of years? So the cool thing is that you don't have to be an expert vertebrate paleontologist to ask important research questions. Your students were it's incredible. Your students were asking the exact same questions that our graduate students are asking in our seminars. So, that's really cool. And I just want to applaud you and Eusebius as well, that I guess I never really thought that kids think that all fossils are large like dinosaurs. And this just expands it expands their minds. And I just think that's really cool. So thank you for all of you. Thank you for your presentations. I want to hear the rest. I'll be quiet.
And with that, last but not least, we have PJ.
Oh, I mean, just 1 second.
Sure.
I just wanted to thank Dr. Holbert for coming in and being with the class. They did enjoy his talk very much, although they're much too shy to ask questions. So that was, I think next time we'll have some pre-made questions for the scientist who comes in. But anyway, thank you very much. I'm done.
Thanks, PJ. Up to you.
Okay, I'm gonna share my screen. I got a little PowerPoint I want to share with you guys. Hopefully I can. Well, let's see. Anyway, I am a media specialist, so I have a lot of classes that come through the media center. So can you guys see am I sharing?
You are sharing. Now we see your PowerPoint. Yes.
So I was very lucky to be able to reach a lot of students in my school. And in a second, we'll talk about our visit with Richard the other day. So basically I did 3rd, 4th, 5th, and a little bit of 7th grade. So I have a few lessons and extended over a lot of days. So I started with this with my elementary kids, with doing some of the videos to introduce fossils. So this was day 1. Now, when they come to the media center, I have to have time for them to be able to check out books also. So it's a balancing act of fitting all of this in. So that's why I had multiple times. I also introduced, of course, the Montbrook on the next session, got them excited, like, wow, we have this in Florida. And this was day 2 when we showed them the slides. Then they all got their Petri dishes and their matrix samples. And I also introduced by using my vertebrae collection out on the tables with rocks, and so they can learn how to identify the difference between rocks and fossils. So that was pretty cool. And they learned that. And so this is just one of my displays that I had up front with, I had a lot of fossils beforehand. And another thing I want to add on to and be able to still do is compare like my Aurora fossil museums with my Montbrook ones that we have now. I had a display in the back of the room also, just showing a bunch of fossils and stuff in the displays. There's my saber-toothed tiger that I had on. It's actually Halloween decoration. So we used the identification sheets that we got and separated everything into the little vials of the samples. Just showing some of this isn't one of my Montbrook fossils, by the way. And I wanted to ask you guys, what is this, by the way? Can anybody tell me? I'm thinking it's from a pufferfish mouth plate, something like that. Can everybody hear me?
I
Yes.
think that's a vertebra, or at least a part of one.
Oh,
Yeah, so no, it's not a pupperfish.
no, okay.
It
I'd
looked
have
like
to
a
see
dolphin.
more sides.
Maybe a shark vertebrae.
No, or, I mean, it could even be whale, some kind of cetacean or dolphin.
Okay. Anyway, just some samples. I took a pretty good ribbing. Try to throw a little humor in there. Found a lot of stingray pieces. So through all this whole experience, now I have students coming into the media center every day because we have a little bit of our matrix that we're still going through. And the kids are still coming in, going through it, separating the fossils. I have my little paleontologist with my 3rd and 4th grade classes. Just some pictures of our displays and the kids going through everything. Of course, they love finding the shark's teeth. That's all they wanted to find. It was a big deal when they found the shark's teeth. But using the identification sheets were cool. I had the microscopes that they got to use to look at things different, their fossils through the microscope, or is this a rock or a fossil? So we started with learning how the difference between rocks and fossils, and then it progressed into Montbrook, then it progressed into, now you guys are paleontologists today. And then I moved on to using the 5th grade standards about adaptations and how those animals at Montbrook, what adaptations they have that have helped them to survive millions of years. So when we had our scientist visit, I had the video, everything going. The book fair gets delivered the same day. So you see up here, these are kids lined up because the sound did not work for some weird reason during my scientist visit. And the kids lined up to type their questions. Or I had another kid who could type fast, type in the questions. There was a huge line. I didn't get the whole picture of that. We had kids on the floor in the media center watching our scientist visit. So I do have to apologize to you guys, Richard. I hope you guys still enjoyed the visit. Our kids loved it. My virtual kids, I was so proud of them asking all the questions. So I'm excited to hear, or maybe not excited to hear your feedback. So one of the things that they're learning with adaptations is, for example, about the garfish and its swim bladder, characteristics that help it survive through the years. So I just had some resources. I love showing the Florida fossils evolutionary of life also videos to the kids so they could see what was in Florida during that time period of when of the fossils that we were finding. So it was a really great experience. I hope to continue next year. Hope you guys don't think I'm a flake because of my scientist visit, but I thought it went really well.
Your visit was great. We had some technology issues, as you mentioned, but kids' questions were flying in, and Richard was, he was rolling with the punches there, and we did what we had to do. So I'm glad they had a lot of fun. I know I did. I won't speak for Richard, but I'm sure he enjoyed it too.
Oh, Iffy, I knew it. I knew he was going to say that. It was crazy. And I'm trying to get these guys up front who were typing the questions, and I had all the kids out in the media center, and then they got a little restless after a while too. So it was, I thought it went well. I'm glad all the kids are excited. And now I have this paleontology club that I guess I'm going to start. I have all these boys, they stayed in after the scientist visit and got to eat lunch with me. And now they want to come
really
in.
Cool.
So, a lot of good things are going to keep happening from this.
Thank you for sharing. Yeah, we're glad to hear the passion and all the stuff happening in your media center. So it looks like a great experience for them, from the literacy aspect to the experiential piece where they get to learn and dig and identify things. So
yeah, all my rocks and minerals books were checked out. Anything about fossils that I had. And that's the great part. I can tie in the literacy and the science together with it, so.
Definitely. So I think Brian's going to stick up. Did we get everybody any questions before we move on to the next slide here? I think Brian's going to stick up a slide that's just going to walk through the breakout rooms. we're going to let you get a little bit deeper into reflection. This is just some of the guiding questions for the next portion. You're going to be split up into grade bands, and we want you to talk a little bit more in depth about the lesson or series of lessons that you implemented or are in the process of implementing, and then what standards, objectives were they aligned with, and really get deep on what do you think worked, what didn't work, what you would do differently next time, where the role of the scientist is in all of that, and what we need to take from that. So I think, Brian, do you need a few more minutes to work
Oh, we're
on?
good. I was just going to add, by no means do we
Please.
expect you to stick to these questions verbatim. You can take the conversation wherever you'd like to go. We just thought we'd throw up a bunch to sort of get you started as some sort of foundation. But wherever you and your cohort and your scientists want to start talking about it, wherever it takes you, that's great. That's excellent.
Awesome.
All right. At this point, I'll open us up. Ours is so funny. Someone drew a portrait of John, and I love it. The kid. Looks like we're still trickling back in. "15 seconds, they'll all be back." Okay, I think that's everyone. Before we move on, I just want to take a minute and sort of reflect on the breakout room. Our group had a really, really amazing conversation, and I was really excited to be part of it. But my question that I want to pose to the group and sort of put out there is, what were some of your takeaways from your colleagues in your group here? I mean, for example, I know that Denise had to teach the whole thing virtually. So Monica was curious, how do you do that? What does that even look like? I know Monica at one point was curious about, if I were going to do this again, I wish I had done a pre-test about this. And then there were also questions along the lines of, I know I want to do this again, but I might not be in this professional development experience. Like, what does it look like to get our own spoil pile and do this again? And what does that process look like in general? So I just really want to open up the floor and see, what were your takeaways from colleagues or reflections, or what do you want to share with us?
Something Mackenzie and Monica said in our group was the pre and post test. And, you know, as teachers, we know that that's important. Of course, I didn't think about that ahead of time. But doing it for your benefit as scientists, because you guys use that, you guys can use that information as much as we can. You know, that's something that didn't even dawn on me to do a pre-test, but for you guys to show the validity of this program, to other people to say, this is why we need the money. This is why we do these things. This is who it helps. That's something pretty powerful for me that I made notes to myself. Okay, next time do this, because you can use that data information. And as far as virtually, I do it all day long. So that's why ours was almost like a well-oiled machine because we've been doing this since the beginning of the year. But definitely don't do this for the first time with kids in the room. Practice sharing your screen and practice doing these things, practice using the same platform. And Maria did wonderfully for that to be her first Google Meet, because she uses Zoom and we use Google all the time. So she did wonderfully for that because they are two totally different platforms. But making sure you're aware of that, and because kids are pretty forgiving, and actually very forgiving when it comes to technology. They'll be like, oh, it's okay, no big deal. And making sure that they have that attitude, and teachers, we have to accept that it's okay if it doesn't work perfectly. You know, they know that we're all learning. And so that's a big thing for us.
I think, Mackenzie, the point you made at the very end of our session was very important. I don't know if you want to share it.
Yeah, I was going to talk. I wanted to let some of the other teachers talk first. The one thing that I kind of made a note of, and we didn't have quite a lot of time at the end, is for you teachers. For a lot of us scientists, one of the very most important thing for us is actually the feedback from you guys. Obviously at the beginning, telling us what you guys want from us out of the lesson, but at the end as well. This PD has actually been great for me because Monica provided so much feedback on how my lesson to the students went and how I performed with them, especially virtually. And maybe how I can improve on that in the future. And sometimes with teachers, I don't always get that. I just get like, oh, like you did a great job, which is awesome. I'm happy to know that I did a great job, but having actual, feedback that I can actually look at and say, okay, this is where I can improve with my science communication skills, with my science education skills in the classroom, especially for a lot of these scientists that are doing this a lot. And this is what they're, passionate about. I think this is such a great program that develops those skills in scientists as well. So maybe building that in, or having, teachers be more aware of that feedback on the back end is just something I wanted to note.
I kind of wanted to add something also. I spoke to one of the people who participated last year, Andrew McIntosh, and he was telling me how it was run without having to do it virtually last year. So maybe a combination of meeting live in person and virtual, depends on what's happening, of course, next year with the epidemic. But, it did sound a lot different than the experience that we had this year, of course, because we had to do it virtually. So that would be something to think about. Maybe even having those people giving feedback. So everybody, we could put something together or thinking about it for the future, virtually or live in person. I don't know if that helps at all, but.
Stephanie, Brian, I don't know if you guys are planning on sharing our slideshows, but I think this would be a great resource too, to just build a library of lesson plans, because they inspire. I mean, the things like Anne-Marie, what you did with your K group blew me away, really. So, and there are some things I would definitely even use with my fourth graders, just, adapt them.
Absolutely. I was also so impressed with that kindergarten level. We'll make the Google Drive so anybody could just upload their file to it if they feel comfortable doing so. And again, we'll include that in an email following today. And again, if you're comfortable, I encourage you to share your presentation. I would encourage everybody to see that portrait drawn of John.
I don't know if he wants me to mail it to him so he can stick it up on his fridge.
Am I the only one who got a portrait, or?
Yes.
Okay.
That I know of. I mean, she was so excited. I'm like, oh.
I love that.
I love the ponytail and the green hair.
I got sent, just as a aside, I got sent a picture last year of, from a mother whose child that dressed up as me for Halloween.
I know they didn't not everybody got to see the picture. So I'll try to, if my, here's his picture. Here's John. And she wrote, she's one of my best writers actually. And she says, I love the fossils the scientists find. So here she is, here's his long hair and glasses. I loved it.
I just want to see if anyone else wants to share a comment. I feel like I've really enjoyed hearing from you all. So if anyone else wants to share, please go ahead.
I have just one thing, if it's okay.
Sure.
You know, at first I was really intimidated. I've always respected scientists. I've always kind of wished I had gone in that field. You know, I wished I was in the dirt digging myself, you know, but so at first I was so intimidated. I was so nervous. I was like, I don't know anything about anything fossils as far as, you know, this goes. And I thought I was going to look really dumb. So I am just very grateful and appreciative of how patient and kind and supportive you've all been with teachers, even though we're not, you know, we're still learning and some of our knowledge is almost on the same level as the kids sometimes, you know. And I just want to say I'm really grateful for that, and that I didn't have to feel intimidated. I didn't have to, you know, I wasn't presenting as though I was a scientist. I was learning from you guys, you know, as well.
Thank you. Appreciate you sharing that. And I want to speak out of turn, but I think we're all still learning. So it's just a journey for all of us. And it's very exciting for us all to do that together. Anyone else?
I have another question for you guys. Do you want us to turn in, like, we had these, the lesson plan format that you guys gave us? I actually like typed out my lesson plans and everything I did on here. Do you want us to, or were you expecting us to turn these in to you guys so you can have those also from this today?
I would say it's not a workshop expectation. However, we would love to have it if you can share it, or if you're willing to share what you did. We would love to have a copy of it, see it, and then ultimately potentially even use it as an exemplar for our next cohort. So again, if you can share it, we would love to have a copy of that as well. So just like last time, we'll include that in the email. If you want to just drop it in that Google Drive, that'll be great.
Also, maybe tell it, because I wasn't sure if we had to or not. So I was like so stressed on making sure I had it all filled out and my lesson plan all typed up, because I thought we were going to be turning them in today. So maybe letting us know next year, don't stress. You don't have to worry about having those all typed up, you know, for the last day. So, or I might have missed something, who knows?
Well, sorry about that. Well, now you have a lesson plan to be proud of.
I got 3 pages here for each lesson, so.
That's funny. All right, if there's nothing else, we're going to take our afternoon bathroom break. Same as last time, it's going to be about 5 minutes. I have 12:33, so we'll come back at about 12:38 or so. And I'll see you then.
Cool.
Looks like some people are still trickling back. I guess we'll get started in another minute or so. If you are back, please turn your camera on just so I know that you are ready, and we'll continue shortly. All right, continuing with the program this afternoon, we are going to talk a little bit about what I saw a lot of people writing in the chat during our little intermission break there. A lot of people are wondering how could I get into the field? And that's what I'm going to talk, or we're going to talk about next, with a little bit about how can you get involved if your students are old enough, how can they get in the field and things like that. So Richard, if you want to talk a little bit about what that looks like for people who aren't scientists, that'd be great.
Sure. So again, to remind you, the vast majority of the people working at the Mount Brooks site are not professionals. So once we return to normal, hopefully in the fall, we typically, particularly on weekends, have sometimes as many as 20, usually more 15 public volunteers. Now, there are certain days if we decide to have a special school teacher workshop or something, the site would be devoted entirely to professional development of the teachers. But let's say on a normal day, that you sign up or register. Our typical age limit is kids have to be 15 years or older up through 17. They can apply, but they have to be accompanied by at least one adult. That adult doesn't have to actually dig, but they need to be there to supervise. But typically the adult or adults do dig with the kid. And so there would be no issue in, you coming as family groups, colleagues, friends. You can tell, again, those of you in middle school who would be 13 or above, that this would be an opportunity for them. And so we don't have yet a set digging schedule. We don't plan that far ahead because you never know when there might be, say, a late season hurricane, which, if we might, say, if we say, we're going to start early October, but then there's a hurricane in late September or something, and we have to really, work the site to clean it up. So we typically, though, so on average, we would be starting sometime in late October and would dig through maybe early December, in that range. We haven't quite finalized how intensely we're going to dig next spring. We vary from year to year on whether we even start as early as late January, or sometimes we hold off till March. And so that's still going to be a discussion between us, what's going to be the best for the museum. But we would have we would definitely going to have a spring and a fall session next year. And so there's sort of two basic websites that people can go for information. There's the Mount Brook blog site, and then there's the site off of our main webpage about volunteering. And so typically for next fall, the information will start to be posted online late August, early September, and you can start reserving dates, usually somewhere between 3 to 4 weeks beforehand, before the season starts. And there is one of the requirements is that everybody who digs at the site, who's going to be a volunteer, has to become an official museum volunteer, which requires a little paperwork. There's forms both for adults and minors, but that's handled by the museum volunteer coordinator. And so it just means that you just can't show up one day without any, pre-registration or anything. And the other thing is that so we always have both digs, both on weekdays and weekends. Whether we would dig both Saturdays and Sundays or just one or the other, again, hasn't been determined. There would definitely be weekend dates for people who can only dig on weekends. And typically, again, it varies, but normally we start work at around 10 in the morning and leave at the site around 4, although depending on circumstances, you don't have to work that entire interval. You can show up later if need be, or leave earlier as need be, but we like people to work at least a minimum of 3 hours. And again, we're more than happy under normal times for just people to essentially do one and done volunteering efforts. So you just come once, but if you want to become more of a regular, that's always welcome, because as you dig more, as with anything, the more you do it, the more you practice, the better at it you get. And so we have volunteers now who have worked with us at a variety of sites for some up to close to 20 years now. And so their skills are as good as, professionals. So we're happy to take anything in between, from, bare people just doing the first time to experienced ones. So again, there's nothing, all I can say to, right now is that we will definitely be doing some Mount Brook digs next year. Some in the fall, some in the spring. We don't dig in the summer. It's just too hot and too rainy. I mean, it's, when you dig, if you dig a hole in Florida, as you can imagine, in the summer, it's going to fill with rainwater. And so Mount Brook is essentially a pretty big hole. And so that's why we focus in on the dry parts of the time. I'll be glad to answer any other questions you might have about that.
Richard, how could teachers stay in the loop and become aware of when digging opportunities are available?
Well, again, I would say that we could probably do a better connection with your group. And so that when I post the web page, then maybe you can flash I don't know if you use Facebook or some other messaging thing to say, information is now out about the dig, because you can probably send this information to all your scientists in every Florida school as a thing. And again, so in the past, we definitely have had teachers come and, normally when you dig, you just dump your sediment, the fossils. You collect the fossils in a separate bag, but then normally the dirt goes on the spoil pile and just is dumped there. But we've had teachers, collect their sediment and to bring back to classes, things like that. You're always welcome to take all kinds of images while you're out there. There's no secret sort of things other than the exact precise location of the dig. So we don't want photos to be posted online that have the exact latitude, longitude of the dig, but so that's a welcome opportunity. So whether, now again, it's a limited number of people. So you're going to have to, particularly if you have a big group, let's say there's, say the teachers from St. Lucie County want to get together and come up as a cohort, maybe the ones that are in the program here and some other colleagues, if you have a group, that's particularly larger, you really have to plan ahead to make sure that there's enough places for everybody. So John, you want to take anything else? Unmute.
I know.
Yeah.
I was just going to say, I think for this group, since we didn't get to do an in-person component to this workshop, which would have been great, we should probably, when we send out emails early on, Richard, to to our diggers, we should probably give this group a little bit of a heads up before opening up to the rest of the general public, just so they can, to give them a little bit of a priority so they can have that experience. So we should send out an email to this group.
Yeah, what John's saying is, again, to sort of maximize the use of our long-term volunteers, is they usually have like a week to 10 days to reserve dates before we open it up to newbies. And so I guess we can consider you guys now part of that cohort. And so you would be able to be in the first, the group, and hopefully, particularly those of you who, might just, there's only one Saturday in October where you're free. And so we can make sure that you're able to get to that date. But that won't happen until, late August at the earliest, more likely early September, when we start really finalizing plans for the fall. And obviously the next spring would be even later.
Any questions for John or Richard?
I have a question. I noticed there was a form that you could fill out to request more bags of that matrix. Can we still do that? Like request some more of those bags?
I'd have to see how John feels about it. Again, it's easy to fill the bag, but actually, again, there's the filling the bag, but then there's taking it back to the museum and actually washing it. Because what you're getting in the mail is like, a tenth of what the original thing is. So it would be, we have a fortune in postage if we mailed you unwashed matrix. So there is a step that takes my time or one of my volunteers' times. And I'd have to see whether, whether it's something that we want to do.
So this came out in the previous discussion group as well. And
Yeah.
I think, so Richard, we can talk about, I think the short answer is I think we can do that. We like Richard said, we need to make a strategy, but I think it might take, we might have to put aside a week or two, maybe a week or something where we just wash spoil pile and then had it reserved so we can send it out when we need to. But anyway, that's just some strategy there. But I do think it's possible.
And the only reason why I ask is because while I was doing this, I had a lot of teachers, of course, walking through the media center that want maybe to do it next year also. So I thought it'd be interesting if they were doing fossils, then they might have their own samples to do in their classes.
So this is just something that we're going to have to talk about as a group, but I suspect we'd probably trend in that direction, but we just need to talk about it and we can send out some more information once we figure that out.
All right, thank you.
And as you might know, those of you who got that stuff, we actually sort of high graded it. So you saw there were bigger fossils and things that John and I, and actually John's family came out to the site, and we sort of scoured because after the rain comes and hits the spoil pile, it sort of erodes away the fossils. And so we scoured those just to make sure that there was stuff in those bags. And so those bags were, the spoil pile bags were a little richer than it naturally is. So we probably can't continue that. But as you say, with the bags that weren't, the real data bags, there's plenty of fossils in there. And so particularly the smaller ones, as you're digging, you just don't see most of those. And so there's plenty of the little sharks and rays and snakes and things. And most of those are just essentially going, we pile them on the spoil pile, and then eventually they get bulldozed away or carted away. And so they're really going to waste. So we'd be glad to put it to good use, but I just wanted to emphasize that it's more of our time than just bagging it up. But
I had a question about that. How do you know where to bulldoze, like before damaging something? Like I'm just curious how you do that.
Well, we've worked enough at this site to know that there's certain levels where the fossils are not present or are very rare. And so, typically though, where we think there's fossils as our idea that, okay, there's probably fossils there, we switch from bulldozers to shovels. When we think we're even closer to fossils, we switch to screwdrivers and trowels, but still dig fairly fast. If we're trying to remove sort of the last meter or yard of dirt on top of where the fossils are. So the closer we're getting to the fossils, the more carefully we're working. And every once in a while, something odd happens. But if there's no way to dig, or I guess there is a way, but if we have to remove 5 or 6 meters of relatively sterile overburden to get to the fossils, it's not worth our time to spend, and particularly our volunteers' time. We ran into that issue at a different site a while ago, and we typically lost a lot of volunteers if they're not finding stuff. So again, when we are, so the spoil pile is stuff that, again, people are in. We can usually tell the difference because the sediment's a little bit different. So the piles that have the barren stuff versus the stuff that people are getting the bones from. And if we know we're going to be doing this regularly, we can separate the spoil piles and say, okay, well, this is the spoil pile for people who are working in the bone producing layers, and the people who are just clearing off the unproductive areas. Yeah, the Dr. McFadden's famous for just going out and, digging 50 buckets of unproductive stuff just to clear down so that the volunteers can get the good fossils. So if we know in advance that that is something that we need to be essentially producing on a, I'll say, industrial scale, we can set up the site and manage the spoil piles a little bit differently as we get going. I don't know if we have a couple more weeks to go in the current dig. I mean, if somebody's really raring to go and they have been vaccinated fully, we are accepting new volunteers, and we have a few weekend dates. We're going, I think there's some places on the first and second Saturday in May. If somebody really wants to get out there, again, you'd have to be fully vaccinated plus the 2 weeks. And we could probably set you up if you really want to go right away. But other than that, if you're willing to wait till next fall, it'll be easier.
I think for the sake of time, we're going to move on, but I encourage you to write any questions you still have in the chat, and then I'm sure John or Richard would be happy to type back and answer any questions you still have at this point. Moving forward, we're going to move to our evaluation. I'm going to type this in the chat too. It is a little bit, I don't want to say longer, but it might take you about 15 minutes or so. So we're going to give you that amount of time here before we start to wrap up today. But again, you should be able to go to that link that I just put in the chat. If for any reason it does not work, feel free to let Stephanie or myself know, and we'll work around it, try to make sure we can get you that link that does work for you. You could ignore this time, 12:55. We went a little bit later today, but feel free to take about 15 minutes or so. We'll come back together and start to wrap things up.
Brian, Jeanette wants to know if we want scientists to fill out the survey as well.
I was under the interpretation it was just for teachers, but if you or Bruce feel differently,
that's fine.
Just teachers, right?
Just teachers. That's the way the survey was designed, correct, Brian?
Correct.
Then that's the way it should be. Otherwise, if we take it, it's not going to make sense. So back about what, 110, 105, 110?
Yeah, 110, if that's enough time, hopefully it should be.
And then we'll do a final wrap up, correct?
Correct.
So good, thank you. Okay. Jeanette? Yeah. They're doing their evaluation for 15 minutes and then I just have to go back and find them.
And again, if any teachers have any trouble accessing the survey, feel free to let me know and I'll send you a new link. But
Hey, I have a question about one of these questions.
Sure.
It says, with what grade and unit did implement your deliverable in the classroom? What is that asking me?
Oh, sorry for any confusion with that. It's essentially asking what grade level did you teach the lesson you developed to?
Okay, sorry, I was like, okay.
Brian, and just a clarification on one question. How might your implementation plan have been different if in-person visits were possible? You're referring to scientist support in person or?
Correct.
Okay, thank you.
Or I guess even would you have designed this differently if you didn't have any students learning virtually? However you'd like to answer that, if completely in person was possible.
Okay.
All right, I think those surveys are trickling in. Most of them have been completed and submitted. We appreciate that. And we just wanted to leave you with just a couple more thoughts as we part ways today. Although we want to be sure to emphasize, and I'll say the last point first, you definitely should stay in touch with us. We hope you do. We highly encourage you once again to jump on our Facebook group if you're a Facebooker, our Twitter feed if you do Twitters, and also follow us on our SEF Spotlight newsletter. Going back to everything you've learned over the course of this long-term PD, we hope that the paleo content that you've been introduced to has enriched your knowledge and enriched the knowledge of your students. And I'm going to place in the chat box, if you already don't know about the request form, we invite you, Brian and I, to request a scientist in any Earth Systems field at any time. That's what we're here for. We're happy to make matches for you so that you can have virtual and hopefully soon physical visits in the classroom. So that's something to keep in mind as well. And finally, there was a really great comment made at some point, I believe in maybe the middle school breakout is where I remember hearing a teacher mentioning other teachers just not being privy to this knowledge and being afraid to teach this content. We invite you to get your teacher colleague friends or district leaders involved in SEFs and tell them about these PD opportunities and about the ability to request scientists, and help us continue to spread the word and grow the program and make it great. So we're excited to have you be cohort number 1, and we're looking forward to working with you some more. I'm going to open the floor real quick to Bruce and John and Richard, if there's anything you'd like to add as we wrap things up today.
I don't need to add anything, and I want to thank Brian and Stephanie who put in a lot of effort in their organizational skills, the students who've also been involved, Mackenzie, Maria, Jeanette, others, and of course the teachers. So this has been, from my point of view, a very successful professional development, and it's a testimony to all of us working together as a team, and I really enjoy that and appreciate it and look forward to more of the same in the future with you all and other teachers as we continue to build out the network. So thanks very much.
Yeah, and everything Bruce just said. And for me, this was really important. It was eye-opening. I'm very much looking forward to doing this again next year. And I'm looking forward to seeing as many of you as I can out at Montbrook when you can make it out there to dig with us.
And I just second all the comments that Bruce and John just made, and to hope to see at least those of you who can make it up to Levy County to join us at the site.
Well, thank you again, everybody. We hope you enjoy the rest of your weekend and stay in touch. Take care.
This
Bye.
has been awesome. Thank you so much.
Thank you so much, everybody.
Yes, thank you very much for everything. That's been awesome. Hope to see you again.
Richard escaped before it's so hard to get, I can't call him. So I don't know if I'm digging with him tomorrow morning or not. I'm assuming yes at this point. Oh well. Thanks, Brian. Thanks, Stephanie. You're still on here. Thanks, Anne Marie. Oh, she's gone.
I'm not.
She's still here.
As I understand it, we definitely have funding for next year to do this again. So.
Oh, excellent.
Yeah. So
sign me up. I'll be back.
This is great. Okay, good. All right, stay in touch, everyone. See ya.
Thank you. Bye, everybody.
Take care.